Welcome to Chapter 4, where we discuss how we learned what we currently know about the human genome. So let's begin. Initially, genetics, of course, was focusing on the identification of genes that cause disease. But how did we go about doing that? Well, originally we would map a disorder to a specific location on the chromosome and then try to find out that location, what gene was involved. When we learned how to sequence DNA in a more rapid fashion, we started what was known as the Human Genome Project, where we sequenced all the human chromosomes and located almost all of the genes. But we still don't know all the specific mutations for most of the inherited disorders, or which gene or genes are going to contribute to them. So the next most important step is gene cloning, which means that we're going to isolate that segment of DNA that codes for a gene so we can study it further. The steps in understanding a genetic disease begin first with, as we've already said, mapping the gene to a location on one of the chromosomes. Then we clone the gene, which is basically just making millions of copies of it, so we have a lot of it so we can analyze it. One of the things we'll do to analyze it is sequence the gene. And then, because we have lots of sequences now, we can compare them with other sequences that other people have put into these large gene banks and see if its function has been identified. And by using these huge gene banks and stuff, this is what we refer to as bioinformatics. Then, using some recombinant DNA techniques, we make the gene product, which of course is going to be the protein. And if we want to discover the protein function, we're going to make out what we call knockout mice. In other words, we figure out the amino acid sequence, make sure that an animal has it, and that this protein seems to have a similar function, same amino acid sequence in other words, and so we inactivate it by inducing a mutation and then see how that animal develops. And so I couldn't resist putting on the little knockout mouse with the helmet and the, of course, fighting gloves. My sense of humor. I know, it's pathetic. So, what else can we do with all this? Well, once we figure out a single gene defect, can we repair it? Can we put the gene back in? And that ultimately is, of course, our goal. So, we're going to map the gene. That's step one. There are two major approaches to this. First of all, genetic mapping. This is looking at the distance of the gene we might want to look at with something that seems to be inherited in all families that have this disease. So then we look for how many times there have been some sort of recombinations or crossovers between those loci, and that will help us to determine the difference or the distance between them. As opposed to physical mapping, which we now can do a lot easier. And this uses cytogenetic and molecular and in silico or computer methods to determine the actual physical location of the disease-causing mutation and the location on the chromosome. But for a long time we were restricted to the genetic mapping until we found out how to do the physical mapping. Even though we have these newer physical mapping techniques, we still frequently have to do genetic mapping or linkage analysis. To understand linkage analysis, you need to understand the principle of independent assortment. This was discovered by Mendel Gregor when he was working, Gregor Mendel, I should say, when he was working as pea plants. Anyway, he found that transmission of one gene is independent of another if they are not located next to each other on the same chromosome. However, linked genes are located near each other on the same chromosome, and so usually if you inherit one, you inherit the other, so they're transmitted together. So you can see this on the lower left over here. We've got 2 A and B linked, and C being independent of those two. So we got the 3 genes A, B, and C, 2 alleles for each, 1 or 2. And as I've already said, A and B are linked. and therefore inherited together, and C is on another chromosome, so whether C1 or C2 is in the gamete is a 50% independent assortment from A and B. Okay, you know that two genes are linked. How do you determine how close they are to each other? Well, this comes about through crossovers. Homologous chromosomes. Remember, you get chromosome 1 from mom and chromosome 2, or another chromosome 1 from dad, I mean. Now, during prophase of meiosis, they line up together, the two ones. And sometimes they exchange DNA. There's a recombination between information on moms and dads. It'll be reciprocal, so no DNA is lost. Typically, we see 3 crossover events for each chromosome during meiosis. So this is a very common thing that happens. And then what will happen is the child or the offspring will inherit then a haplotype, the combination of alleles on each of the chromosome. Now these recombination events, however, will change the haplotype. So without a crossover, you get either A1 and B1 or A2 and B2. With a crossover, however, you can get A1 with B2 and A2 with B1. Double crossovers may actually restore the original haplotypes, but then you wouldn't know that they had actually happened unless you had documented it before. This figure kind of gives you a nice little summary of just some very simple crossover things that can happen. So if you look at A over here up on the top, you can see that there was no recombination between them, so they are inherited as A1B1 or A2B2. Okay, so now in part B, however, we have some crossover events that occur here so that you exchange A1 and A2. So now you can not only get A1 and B1 and A2 and B2, you can get A2B1 and A1B1. Okay, because you got the pink on the blue and the blue on the pink. And then here, if we go to C, now we have a double crossover event, which can then actually restore it. So it's A1, B1, A2, B2, and we're back together. And the only way you're going to know that this C has occurred is, of course, is if you sequenced it and all this sort of thing. Which takes us to the whole concept of recombination frequency. So crossovers are really more likely between loci that are far apart on the same chromosome rather than when they're close. So the distance is determined by how frequently the crossovers occur within a family. So this is, of course, the recombination frequency. Now, if the recombination between the genes A and B only occurs about 5% of the time, then that indeed is the recombination frequency, 5%. So it's not occurring very often, and it means that the genes are very close together. That means 95% of the times you will not get a recombinant, they will be non-recombinant, and they'll be inherited together, and therefore they are linked. And that was where we get this linkage analysis. Now these kinds of studies are great if you got a really large family or you're working with organisms such as fruit flies or mice where you can have large numbers and be able to determine this sort of thing. But what are we going to do if we want to map the genes, of course, in humans? Well, what we have to do is look for markers. So markers are simply just polymorphisms, okay, alleles that mark the chromosome with disease genes. So these linked markers can be used for early diagnosis of genetic disease. They're linked to the disease because they're always inherited along with the disease, but they are not involved in the disease cause themselves. Now, crossover rates, of course, can vary. We find that crossovers are 50% more common in females during meiosis, and that there are some areas that are what we call recombination hotspots. And this is particularly true for the ends of the chromosomes, the telomeres, where the recombination can increase tenfold. So now your distances, of course, are going to be altered. Well, let's look at an example. neurofibromatosis or type 1 NF1. It's a common autosomal dominant order as far as these go. A mild form is just the cafe au lait spots and a few little neurofibromas. They're benign tumors, of course, of the peripheral nerves. The severe form, of course, everybody thinks of Elephant Man, if you ever saw the movies or anything. Now you have literally thousands of these you can have. Optic gliomas, scoliosis, and of course other problems as well, including learning disabilities and hypertension. So it's a very large gene, the neurofibromatosis gene, or NF1. And 50% of the cases are going to be new mutations. So capital N, of course, is the dominant mutation. A little n is a recessive or normal gene. Okay? And it will have 100% penetrance. So here's a cafe au lait spot, and here are the neurofibromas. I've got them. I don't know about you. They come up more as you get older anyway. So let's see an example. In this case, what we're going to be looking at is a marker known as 1F10, and it's going to have two alleles, 1 and 2. Okay, and we're going to say one of those alleles, 1, is associated with the disease gene NF1 and are linked. Okay, so that's our hypothesis. NF1 and 1F10 are linked. So the linkage phase is just talking about the arrangement of the alleles on the chromosome. Anyway, so if we look at our first generation, okay, so the mother and father, the husband has the disease because it's colored in, okay, and he has the genotype 1 and 2. So he has the 1 allele and he has NF1, so that he has neurofibromatosis type 1. Over here you can see that the mother's normal. She's got the secondary allele and she does not have the 1, and therefore she does not have disease. So now we go to the second generation, same as the first. We've got a female though who's affected, so now we know it's autosomal dominant. And again, 1 is linked with 2. Okay, but she gets married to a normal man, and a recombination occurs. You have one child where you have that who is affected, which is right here, child number 6, who has the 2 but also has the disease. So one child with recombination, 1 in 8 events, we have a 12.5% recombination. And what we're looking at here is just what would be a gel. And if we take a look at number 6, of course, we're going to see that it has both 2 alleles, and yet it also has the NF1 gene, so a recombination had to have occurred. Now, since the recombination frequency is less than 50%, we say that the loci are linked. However, we would really like to have more data than this. And of course, having 8 kids is pretty good for a human family. This leads us to a statistical analysis that's referred to as the logarithm of the odds, or the LOD, or LOD scores. Okay, and it's used to just statistically evaluate the probability of a linkage result. Is it real, or is it due to chance alone? Okay, so we're comparing the likelihood that 2 loci are linked at a given recombination frequency, and that of course is theta, and the likelihood that the 2 loci are linked or not equals 50%. So they're either going to be linked or they're not going to be linked. There's a 50-50 chance. Okay, so then you use the pedigree data to calculate likelihood ratios at various recombination frequencies. So likelihood ratio is the likelihood of observing the pedigree gamma if the theta is equal to 0.1, or if it's 0.4, 50%. So now we're looking at a 10%. That's all the difference is. Now, for easier reporting, we take the common logarithm of the ratio. So we're taking the logarithm of the odds, hence the LOD score or LOD score. And we have found that if the LOD score is greater than 3, then the likelihood of the 2 loci linked is, because it's a logarithm, 1,000 times more likely than they're not linked. And if the LOD score is less than -2, then the odds are 100 to 1 against linkage. So now we can take an NF1 pedigree, and we again have the family type for the 1F10 marker. The male in generation 2 got the NF2 from father along with the NF1. So, that's fine. But our hypothesis then will be every child from the second generation male homozygous will have the disease. Okay? Father can transmit N1, N2, or the N1, and mother can transmit N2 because that's all she's got. So heterozygotes will be normal for the F1, F10. Homozygotes with the 1F10-2 will be abnormal, and this is what you're seeing. So determining the law score then is the hypothesis of close linkage equals 0.0. If father 2 can transmit N1 or N2 or capital N2 or little n1, probability of each event is 1 over 2. Thus, the probability that 5 children have the genotype is 1 over 2 to the 5th, or 1 to 32. So that's the numerator. If they're not linked, then it's a 50-50 chance, right? So the the father in the second generation can transmit one of 4 genotypes, and the probability will be 1 to the 4 to the 5th, which is 1 in 1024. So that's the denominator. Now you're beginning to see it. So 1 over 32 divided by 1 over 1024 equals 32. The log of 32 is equal to 1.5. So what does that mean? Well, unfortunately, it means we need more data. And so then you would go and try to find additional families. Now, the linkage analysis got a lot better once we got more of what we call a hap map, a haplotype map. And that is with all these markers all over each of the chromosomes. So this was something we really wanted to do so we could locate these disease genes to a specific chromosome. So we are typing members of a pedigree for marker loci in each of the chromosomes in order to do this. And then we test for linkage between the disease gene and each marker. This can be tedious, and it was for a long time. Anyway, the human genome is large, so you need to test dozens or hundreds of markers before you find a linkage. So, and it took years, but we found cystic fibrosis, Huntington disease, Marfan, and of course NF1 this way. And this is what a linkage map looks like. Now that we've sequenced the whole genome, we have thousands of new markers, making it easier to find linkage. And usually these markers are repeats or mutations, so that PR should be mutations. Now we can typically map a new disease gene in a few weeks or months. So what are the useful marker loci? Well, they are codominant, so we detect homozygotes from heterozygotes. They're numerous, can get close linkage, okay? They're highly polymorphic, makes the parents heterozygotes and easier to establish linkage phases. And these, of course, are linked to some of the polymorphisms that we're talking about. And then highly polymorphic markers allow observation of recombination within a family. So this is what we love to find. We don't always do, though. Then there is the concept of linkage disequilibrium. What it's saying is that certain alleles association of 2 different alleles at a loci are non-random. In other words, the haplotype consists of a marker allele and a disease-causing allele, and this is a mutated gene, and this occurs more often than chance allows. So the same diseases may be linked to 2 different markers in this sort of situation. So disease-causing mutations occurred multiple times in the past is one possibility. In time, crossovers also occurred, so there was a non-random association. Or the linked marker may associate within a family but not between families, and we do see that as well. Linkage equilibrium, then, is a statistical independence of a gene and a marker so that they are not linked and they sort independently. So linkage disequilibrium is going to be a non-random association of a marker and a mutation, And the linkage equilibrium will be these two, I guess the best way of saying it is these two markers are independent. Well, we got to have an example. And this example goes back to the myotonic dystrophy. Remember that repeat expansion one. The locus is on chromosome 19. And we have marker A was about 5 centimorgans away. And that means it's a fairly long distance away. But marker B is less than 1 centimorgan. Each marker has 2 alleles, and you have 4 possible combinations at the 2 loci. So if you take a look at this, the way it's set up over here on the side, here's marker A, here's marker B, and here's the disease or myotonic dystrophy mutation right here. Okay, so you can see marker B is closer than marker A. And so through time, recombinants occur and distributing the DM mutation on other haplotypes. And so here's the population with the disease. We've got 1 and 2, 2 and 2, 2 and 1, 1 and 1, and normal control have 1, 2, 2, 2, 2, 1, 1, 1. So they have 4 possible combinations for all of them. What we see with linkage disequilibrium is that it really is a function of the distance between the loci. So we used it to infer the order of genes on chromosomes because of these recombinations. And they would occur over dozens of generations and also influenced by things like natural selection and genetic drift, because sometimes a mutation will confer survival. This is true for bubonic plague and HIV. There were these people in this town in the 1700s in England. Bubonic plague broke out. They voluntarily quarantined themselves. 50% of the people died. 50% survived. Descendants are there 300 years later. HIV comes along, and some of them get exposed, but they never get sick. Why? Because HIV and the plague bacillus, a bacterium, used the same receptor on the surface of cells to get in and replicate, and the ones that survived had a mutation. And they passed it on to the children, so those people were immune to HIV, which is also partly why they survived the bubonic plague. So you can see how that would be, of course, an advantage. Then you have to realize that you have associations in populations, and they may not be true linkages. If there's a linkage, then the position of the loci in the chromosomes, and we're talking about our close and specific combinations of the alleles, will then be linked and transmit together. If it's just an association, it's going to be a statistical relationship between two traits in a general population, and it'll be a little bit greater than random chance, but it's still a probability. Example of this is ankylosing spondylitis and the HLA protein B27. Now, I don't know how much of you are familiar with HLA, but they stands for human leukocyte antigens, and it has to do with the immune system, and it is proteins that mediate graft rejection. Well, anyway, but that's not their true function. This association with ankylosing spondylitis and HLA-B27 is a population association without being a linked loci, because not everyone with HLA-B27 get the disease, but 90% of those who do have HLA-B27. Well, it turns out ankylosing spondylitis is an autoimmune disease, and since HLA-B27 may present self-antigens to immune systems inducing disease, it may contribute to the cause. So it's not linked, but it is associated. And this gives you an idea of what happens with ankylosing spondylitis. Here's a normal spine, and with time these things get fused and get very hard, and they can't turn their head without turning the whole body and stuff like that. It's really nasty, and you can see how it progresses with time. It's not something, let's put it that way. So then we get to talk about a physical mapping. Well, that assigns specific locations to the markers and the disease genes. Uses a variety of techniques. We look at the chromosome morphology with banding. And sometimes chromosomal abnormalities help us pinpoint something. We also look at dosage mapping with deletions and duplications to help us pinpoint it. In which case, a deleted gene and only half of the normal, half of the protein is present because one is deleted, or duplications, you see too much of the protein. So that can help us. We notice that there is either a duplication or a deletion, and we recognize a particular enzyme that might be associated with it. So that's one way of mapping. Positional cloning, however, is going to be a little bit different. Now we're going to be talking about first functional cloning, where we know the function of the protein and we use the amino acid sequence to get the DNA sequence. This is what we did originally, but now we get the DNA sequence right away. We make a probe. Now a probe is going to be a complementary copy of the DNA sequence, and you screen chromosomes for the gene location. Positional cloning, you already roughly know where the gene is from linkage analysis, and then you try to find it or specifically identify it. Now that we have the whole human genome sequence, we can do this. We can scan databases for genes in the area, and once possible genes are found, then we clone them and determine their function and see if they really are the disease gene. However, you got to remember, most of the human genome is what we refer to as non-functional. In other words, we don't know what the function is, and it doesn't really cause disease as far as we know, except sometimes with these repeat expansions. So we've got to develop proteins that scan for the coding sequences, and this is where these computer whizzes and stuff who come in with the bioinformatics, of course, are exceedingly important, is what I was trying to say with this whole analysis. When we talk about cloning, we're really talking about making a recombinant DNA molecule, and this is really genetic engineering. So let's define these terms. Genetic engineering is just manipulating genes in the lab. Okay? Clones are identical copies of a sequence of DNA. Usually we want a particular gene. And we engineer clones so we have enough DNA to sequence the DNA, study it, generate proteins from the genes. In order to generate the clones, though, we have to place our isolated gene into a piece of DNA that can replicate. When we do that, they're referred to as recombinant DNA molecules. What can we use as a vector? Well, anything that will replicate inside a cell. So a bacterium can use, and we can use a virus that will replicate in a cell, a plasmid, which is a smaller piece of replicating DNA in bacteria cells. We can use yeast chromosomes in insect cells in order to replicate the genes, and then these cells will also, in many cases, make the proteins for us. So once the recombinant DNA is placed into the cell, when the DNA replicates, it makes copies of the gene in DNA, and those are the clones we're looking for. So the function of gene cloning is, first of all, to isolate a segment of DNA that codes the protein. All right. So we got to isolate DNA from the cells of individuals with the disorder. What's the easiest source? Blood. And what are we getting there? The white blood cells. Why don't we use red blood cells? Because they don't have a nucleus. So we need DNA, remember? You only can find DNA in our human cells. Well, the easiest ones to get, of course, are our white blood cells, the ones that have nuclei, as you see where my little marker is going. So then we break open the cells so we can isolate the DNA. And the problem is, as you see over here on the lower left, it is very long and stringy. So how do we get smaller fragments? Because remember, we only want that one gene that we want to study. Well, what we use is sort of molecular scissors. And they're enzymes that will cut DNA at specific sequences. So we call those that they restrict the DNA, they make them smaller, so we're calling them restriction enzymes. And again, they recognize a specific sequence in the DNA, and wherever those sequences occur, they make a double-strand cut. And that produces a whole bunch of smaller fragments of different sizes. So here's an example. And what you do is the cut is, as you can see, is kind of staggered, but it's at the same spot on both DNA, strands of DNA, but because of this we have overhanging ends, sticky ends that will base pair back up, as you can see, because they're complementary. Then if we isolate all the fragments that we generate and stain them so we can see them, the separation is based on size, so the smaller ones migrate faster, the larger ones stay up here, and we can assess the sizes of these and try to cut out. If we know the size of the fragment that we think the gene is on, we can actually cut these out and then insert them into a vector. In step 3 then, what we're going to be doing is actually making the recombinant molecule. In order to do this, you take your foreign DNA and you cut it with a restriction enzyme. And then you take your vector and you cut the vector with the same restriction enzyme. And you get these overlapping sticky ends, which are complementary. So then you add an enzyme, DNA ligase, that will add the foreign gene into the vector, and then you can introduce the vector into a host. So vectors are pieces of DNA that replicate in there, and when they replicate they make copies of the gene for studying, and you need lots of copies of the gene to sequence it. And then of course you can use some of the genes to produce large amounts of the protein as well. Well, let's talk about the cloning vectors just in a little bit more depth. Plasmids are really useful because we can put those in things like bacteria like E. coli, and they replicate. They're very easy. Viruses are good too because we can put some genes in bacteriophages and then they'll infect bacteria, or we can put some genes in animal viruses that will then infect animal cells, and it all depends on what we're using. We can also use yeast artificial chromosomes. They're called YACs. We actually genetically engineer them and they'll replicate inside yeast cells, and we can make, like human proteins and stuff in there. And then bacterial artificial chromosomes can also do it. And why do we have so many different cloning vectors? Because sometimes we want to put more or less sequences in there. And so some of these YACs and BACs allow us to put larger amounts of genes into these vectors. And then there is the PAX. So this is a bacteriophage. It's an artificial chromosome that allows cloning of very, very large fragments of DNA. And so this shows you a bacterial artificial chromosome over here on the side. And all these little numbers and letters here, like APA1, is referring to different restriction enzymes that will cut it at specific spots. And then you can insert your gene and then put the vector in the appropriate host cell. This way is actually how we make genomic libraries. So you get the white blood cells from a person. When you take their blood, you're getting all of their genes. So you're getting every chromosome and everything. And so what you do is you cut them all up with the restriction enzyme, so you get all these different fragments, and then you insert them into the vectors, and then put them in the vectors, allow the vectors to infect cells, and you spread them out, and you can test different clones or colonies for the gene that you're looking for. And so you actually have what we call a genomic library. In the previous slide, we described how you would go about making a library using genomic DNA. But what if you just want to know what was being synthesized, or which genes were being synthesized into messenger RNA and then being made into proteins? In other words, which genes were being expressed? Well, you can do that by isolating RNA. So first of all, you would isolate the RNAs, it says here, and don't worry about the primers, but it's kind of easy to get them out because all messenger RNAs in our cells have these long rows of A at the end, so if you've got something with a long row of T on it, they all stick. And then you need to convert that RNA to DNA so you can put it in a vector. And very nicely for us, retroviruses produce an enzyme known as reverse transcriptase. And it can take RNA and convert it into DNA. At this point, you can put it into a particular vector, and the vector in this case is a bacteriophage known as lambda. And so you put that, you make all these different lambdas that way, and then you can infect the E. coli. And then again, you put it on a plate, and now you have all these colonies of E. coli that have lambdas in them that carry different proteins, and you just have to screen to find out which one you want. Now, all of this came about, the ability to do this, because of the very first technique for identifying genes in the DNA known as southern blotting. And the reason it's called southern is because the man who invented it was E.M. Southern. Anyway, in 1975, he knew that if you denatured DNA, so you separated the two strands, you can have them come back together because of those complementary bases. So, you would denature the single-stranded DNA. There we go. And there's the two strands, and they're complementary, as you can see. And then if you separated them usually by heating, and then if you cooled slowly, they come back together like you see there. So, well, that sounds wonderful, but how do you know what's there or anything like that? How do you know that it's the gene you want? So the steps go like this. After you separate the different size fragments from the gel, so remember you're going to isolate your DNA, you're going to cut it with restriction enzymes, separate them on the basis of size in a gel. You actually transfer all of those DNAs onto something called nitrocellulose paper. And this is just a type of paper that will bind DNA very strongly. Then you denature it so that it's single-stranded. And to find where your gene is, you add a probe that is complementary, labeled with a radioactive element. Okay? What'll happen is you add this probe in excess. Right. So when you cool it down and let it reanneal, when you neutralize, in this case what they did to separate it was put it in an alkaline solution, then neutralize it back down to like 7 pH. The DNA and the nitrocellulose, well, H bond means hydrogen bond with the probe because the probe is in excess and it's complementary. And then because it's radioactive, you put it against X-ray film, you can see where the bands are. So what you're doing is you're making a double-stranded DNA composed of the probe and the DNA from whatever sample you got that from. So this is called a hybrid, and hence the name hybridization, Southern by the man's name. So, as I said, you place the extra nitrocellulose against an X-ray film, and as you see here, you can see, and of course we also separated them so that we could see them in different lines. And this is how we get these bands here that you see, and you can see what different size genes or the allelic forms that the various people in a family inherited. And this shows you, we saw these bands before with the example about linkage analysis and everything. And so you can see we go through up here the restriction family, and then you would use the probe here, blotted on there, and you would be able to actually see these bands. And that started way back in 1975. Of course, we have a lot more sophisticated techniques now, but all of these techniques are based on the fact that DNA is complementary. And when you separate the two strands, if you've got a complementary strand in there, it will reanneal, come back together. And if it didn't have that, we couldn't do that. So all of these new techniques are just an extension of the fact that DNA is double-stranded and it's complementary. And that's why we talk about the chemistry of DNA first, because then understanding all of these testing techniques becomes more understandable. DNA sequencing. The first techniques, however, had nothing to really do with hybridization, however. Of course, DNA sequencing is determining the linear sequence of bases, A, G, C, and T, that make up a gene. And the first method that really took off and was able to be automated was the dideoxy method of DNA sequencing. This was invented again by a scientist by the name of Frederick Sanger. And it makes use of the fact that if you add, instead of deoxyribonucleotides, dideoxynucleotides, you stop the synthesis. So they call this chain terminating dideoxynucleotides. And it's similar to the normal deoxynucleotides, except they're missing a single hydroxyl group, so they're dideoxy. They've lost 2 hydroxyl groups. And as a result of this, it can't form a phosphodiester bond, and the growing DNA strand stops. It works like this. You get 4 test tubes, each one, and you label 1A, 1G, 1C, and 1T. Okay. Each tube is going to contain that single strand of DNA you want to sequence. DNA polymerase, because that's the enzyme that it synthesizes DNA. You need primers, so there is a little bit of hybridization going on. Remember, DNA polymerase needs a primer in order to get started. You need each of the 4 regular deoxynucleotides in all 4 of those test tubes. However, a small amount of the dideoxynucleotide labeled on the tube is added. So adenine dideoxynucleotide is in the tube labeled A, and guanine dideoxynucleotide is in G, and so on. So whenever the dideoxynucleotide is randomly added, and since the dideoxys are in a lot lower concentration than the deoxy, synthesis will stop at different times in each of these little tubes, and generating a series of fragments of different sizes, but all ending with the same dideoxynucleotide. So all of the fragments in the tube labeled A end with A. Okay? So the fragments in each tube are then separated electrophoresis in the base of size. They're run side by side in a gel, which you can see over here. Each band ends with one of the 4 bases as labeled in the test tubes. And then the sequence was simply read by starting with the smallest and moving their way up. So if you look at this, you would read, I think this is C, G, A, C, and then, or maybe this one. It's easier when you actually have it in front of you. But typically what we would do is sit there and one person would read it off and the other person would write down the sequence. Of course, that was before we had all the great computers. Now, the technique has been automated using primers that contain a fluorescent or chemiluminescent detection system, so we don't have to use all those radioactivity anymore, which has made us all very happy. And again, instead of getting the bands that you see over here in the lower right, we're going to see peaks in the next slide. And here it is, and the different colors each represent a different base, and all you have to do in the computers now, even just put the sequence down there, and you can then put that right in a database nowadays and store it. So it's a lot easier, and this is all automated. Now, a more rapid technique, instead of using a gel and these big plates, uses a capillary tube. And both methods allow for the simultaneous analysis of up to 96 samples, so that's pretty good. Unfortunately, I think the bases analyzed could only be about 500 to 600 base pairs long. So, but that was good enough for our Human Genome Project, which started in the 90s and they felt would end this year, 2015, but they did better than we thought because we got next generation sequencing. These automated methods use more of hybridization type techniques, can sequence an entire human genome, 3 billion base pairs, which took us about 15 years to do the first time, in 4.5 hours, and that's going to get shorter. The cost of this is already around $1,000, so personalized medicine will become more and more prevalent as we understand all of this data that we're generating. Right now, we are just in the infancy of interpreting all the results that we're generating. So the computer analysis of DNA sequence is again part of bioinformatics. We refer to this now as in silico research. And the computers will scan for stop and start codons or intron-extron barriers to help us identify genes. They can recognize patterns typical of specific proteins. Is this a transcription factor? Is this a transmembrane protein? And then we'll go and compare your sequence with other sequence databases. It's just amazing what they can do. So after a gene is isolated, computers can screen for mutations in sequence in other families because we've been generating all this database. And so now we can compare DNA of unaffected parents with affected children. We see a lot of harmless polymorphisms in both, but harmful mutations will only be seen in the affected. And so we can find these things so much faster than we used to in the past. Well, okay, let's say you've got your gene and everything. What do you want to do now? Well, you want to make some of the protein. And so you're going to put it in a recombinant molecule and into a host cell and let the host cell produce the protein for you. This is, after all, how we produce human insulin and give to diabetics. But you got to use the right host cell, and that's going to depend on the protein. So you also want to isolate and purify the protein for further studies. You can determine the protein structure, which sometimes helps us understand function. We do that with X-ray crystallography. Then we can develop antibodies to the protein. Again, where is it located within the cell? If we add an antibody that blocks its function, what happens to the cell? So that helps us determine function. And then, of course, we'll induce mutations in a gene to make a knockout mouse and then see if the mouse even develops. Or if it does, what kind of disease does it get? So that helps us understand how these proteins interact and then lead to disease. And finally, typically what you do is also, if you're going to do a knockout, that's a subtraction technique. You want to also do the addition experiment where you add the gene to mice who do not have the gene, and sometimes that's your knockout to see if you restore whatever's been missing. And these are known as transgenic mice. So far, you've learned how we studied one gene at a time. And that's the way it was for a long, long time. But nowadays what we're doing is we're studying multiple genes or set of genes that influence a particular pathway. Let's say the electron transport system or the Krebs cycle or anything that would cause a response or a change in the cell's environment. So if we're looking for genes and then looking at the DNA and the study, this is known as the genomics. And this is what you hear about more and more. If we're looking at all the messenger RNA that is being made at one time, then we're looking at all transcription. So we refer to that as the transcriptome of a cell. And then if we're looking at all the proteins that are being made under certain conditions, we're looking at the proteome of a cell. So whenever there has an OME ending to it, we're looking at everything all together at once. And all of these techniques can just do that. Literally look at thousands of DNA, genes, RNA, and proteins in a single experiment. Let's see how these tests were developed for messenger RNA, in other words, the transcriptome. Well, the first test that we had for messenger RNA was like southern hybridization, but we called it northern blots or hybridization because we isolate the messenger RNA, separate that in a gel, and hybridize it, however, with labeled DNA probe. Okay, because you can make RNA hybrids just as well as you can make DNA. But that only really allowed you to look at a few genes at a time. And this is kind of what you would see over here on the left. But now we have DNA microarrays, if you've ever heard the term DNA chips. And what you'll do is simply isolate the patient DNA or messenger RNA, and then you expose them to all these little oligo, meaning a small number of nucleotides, on this little chip. It's actually a very small, it's a glass slide in truth, that have hundreds of these single-strand oligonucleotides that are complementary on there. And so simultaneously you can look at 5,000 to 10,000 different genes. And this is just showing you how you prepare. You have normal and you have abnormal, and then you put them in all these different wells. Here's the little chip over here on the side, and you can see that it's going to be a little bit more complex than that, but from there you can literally look at 15,000 proteins at a time. It's really cool. But of course, this is one of those things where we're still trying to figure out what all this information is really telling us, and it's going to take a while. Well, what about the proteome? Well, we started out with two-dimensional gel electrophoresis, and that's what you're looking at the bottom of the slide. Basically, we'd take proteins from cells that had been cultured in media with radioactive amino acids so that they would make a dark spot on an X-ray film. And then you'd run electrophoresis in two directions. So basically, instead of getting bands, now you got all these little dots indicating individual proteins or very closely related proteins. And so again, you'd incubate the gel with the X-ray film and you get what you see down here. Well, then that was kind of tedious because you'd have to pick one spot and see if it was in a cell that you had treated differently, and if it wasn't there, then you would have to try to find out what that was and all this sorts of fun stuff. So that was pretty tedious. So now let's go to the next slide and see what we do. Well, just like with DNA chips, we can have protein chips. And this particular slide here shows you a figure that on the top here, what we're seeing is a DNA chip again. On the bottom, this would be an RNA chip, and we can do that to look at the transcriptome. But for the proteins, this is here on the bottom, we attach the proteins to a chip, and then we have antibodies, which it doesn't say here, but they're all labeled with these different colored fluoroprobes, and then we can detect them. A computer would detect them, and it would tell us which protein was being made, and because the antibody would bind. And another one that is useful as well is if you combine liquid mass chromatography with mass spectrometry. And basically this is kind of what's done a little bit for detecting all of the proteins and giving us a chance to, we get all these different peaks and everything which will correlate with different types of protein. And this is something that's being used a lot too. I don't think though, however, it's going to be as frequent in patient analysis as it is in research, but we'll have to wait and see. I've been wrong about some of my predictions in the past. Of course, I won't tell you that, which ones. All right, so types of DNA is what we're going to end with in this, because we've already seen that there are these repeats that can expand that can cause disease. So you need to understand that actually the single copy DNA of genes and stuff does not represent that much of our genome. So single copy DNA is only by 45%. This is what is encoding proteins or RNA molecules. As it says, only 45%. Repetitive DNA is just about everything else. Sequences are repeated throughout the genome on different chromosomes, and they're repeated thousands of times. This is more than half of our genome are these repeats. So now you see why I say we don't really understand why they're there, but there's probably some good reason that God put them in there. We just don't understand it yet. Some dispersed repetitive DNA is scattered singly throughout the genome, and that actually is the bulk of it. And then satellite DNA are just repeats that are really clustered together. But we're going to talk about that satellite DNA because those are the kinds of clusters that can expand and cause problems. They got their name, satellite DNA, because they were easily separated from the rest of the DNA when you would put it in a density gradient in a centrifuge. Again, instead of separating it by mass, you're separating by density, which includes both volume and shape. So we found out that by doing this, that satellite DNA makes up 10% of our genome. And there are different types of satellite. There's alpha satellite, and these are tandem repeats of about 171 base pair, and that can be for several million base pairs. Then we got mini satellites that again are tandem repeats, but they're smaller, anywhere from 14 to 500 base pairs, hence the fact that they're mini satellites, whose total length is less than a single alpha satellite. And if you want to get even smaller, we have the microsatellites. And these are very small. Repeat units are 1 to 13 base pair, and usually only a few hundred long. Now, mini and microsatellites vary from person to person. And we don't exactly know the mechanism for it, but we can see that in some cases it can lead to disease. So what about all this dispersed repetitive DNA? Well, there's two types. Short interspersed elements known as SINEs. They're anywhere from 90 to 500 base pair longs, and some of the more common ones are called ALU repeats, and they're about 300 base pair longs. Basically, they're a family of genes. A million of them are scattered throughout the genome and make up, again, about 10% of our human DNA. And then there's the long interspersed repeats, or lines. They're larger and can be up to 7,000 base pairs long and can generate copies of themselves and insert copies into other parts of the genome, disrupting a gene. And that, of course, can lead to disease. So this whole unit, of course, is about the human genome and stuff. So you have to understand that our genome is not just made of genes that code for proteins or RNA molecules. There are also these repeats, and as we saw with patterns of inheritance, if they increase, in some cases they can lead to disease. So nowadays, when we talk about human disease, we talk about something we call now systems biology. And this is an integrated approach to the study of a complex biological phenomenon. So what are we doing? We're incorporating DNA, messenger RNA, and protein data. So we're incorporating the central dogma of biology so that we can understand their interactions and understand how mutations and environmental conditions then can affect not only the function of the cell, but then the individual. Remember, we're a multicellular organelle, or I should say not organelle, multicellular organism is what I wanted to say. And that means if enough of our cells die, we die. So here's a single fertilized cell, and leading, of course, to a human being. And then I don't know about you, but that's one of the, I think, the greatest mysteries on our planet. So that's it for Unit 2. I will see you next week. Bye.